In the last section of our genetics chapter, we're going to talk about genetic technologies, things we can use genetic technologies for. Things like identifying, in this example here, we've got the tomb of the unknown soldier. We identify some remains left at a scene, remains at a crime scene, identify a body, all kinds of things, criminal investigations, where we can look at the DNA. We can do what's called DNA fingerprinting, which allows a researcher, which allows a crime scene investigator to look at a DNA sample left at a scene and run what we call a gel electrophoresis, or use a gel electrophoresis in order to match up an individual with some biological remains. Now, how this DNA fingerprinting is going to work, here's some evidence left at a crime scene. So, crime scene investigator is going to take a sample. That sample is going to have some cells in it, so a few blood cells at the bottom. We can collect those cells, and in those cells there will be some DNA. Now, we use some enzymes that make some SNPs on the DNA, and depending on the individual, the SNPs are made in different locations. This is the gel electrophoresis. So we put some samples of those cells in the machine, we use a current, and the current carries those sections of DNA from one end of the machine to the other, and we get this unique banding pattern, unique for each individual. So this is, say, suspect number 1 here. This is suspect number 2 here, and this is a sample left at the crime scene. So what it allows us to do is forget about this person over here. That sample didn't come from that person, and then maybe focus in on this person over here. The banding patterns, you notice, match up identically. Now, some cool sort of science fiction-y kind of things that can be done with genetic technologies would be transgenics, where we can take genes from one organism and we can transfer, or we can splice those genes from that one organism into a completely different organism. So again, the level of complexity in Science 30 is not the same as what we would have in Bio 30. So in these examples here, you can take a gene from, I believe in this case it was jellyfish, so bioluminescence. They took that gene from a jellyfish, inserted it into some rats and into the germline layer, into the sex cells, which would become these rats, and now some of these mice or some of these rats bioluminesce. They sort of glow in the dark. Now, it didn't turn it into a jellyfish, it just incorporated that one section of DNA from one organism into this organism, and then with protein synthesis it will express that trait. There's another cool example of a goat where we've merged some spider silk genes, or the gene from the spider to make silk, into a goat cell, and then that goat cell here, when it is allowed to grow, you now get a goat as an adult that's going to make milk that will have the protein for making spider silk in it. So the goat didn't come out with 8 legs, the goat just is able to make a protein that came from that same gene that came from the spider to make spider silk. So that would be called transgenics, and we can also call that genetic engineering, where we modify an organism using some genetic techniques in order to change the genetics or maybe incorporate genes from one organism into another. This leads to some ethical issues with what we call genetically modified organisms, or GMOs, which you likely hear about in the news, where food has been genetically modified to have certain characteristics. Trust me, most food that you wind up eating nowadays has been genetically modified in some form. Corn, even the strawberries you buy at Safeway have been genetically modified. They're not supposed to be those big, huge red strawberries. If you taste a organic farmer's market kind of strawberry, or if you have a if you have a strawberry plant in the backyard and you taste those strawberries versus the big ones at Safeway, you'll know what I'm talking about. The small little ones taste really sweet. The big, huge Safeway strawberries, grocery store strawberries, don't have the same kind of flavor because the sugar has been sort of spread out over a larger area. They look better, but they don't have the same kind of flavor. Now, here's some examples of how we make products like insulin. So, for example, here's a human cell that has inside the nucleus the genes, the chromosomes that contain the gene for making insulin. Now, for people who are diabetic that can't produce insulin, we can now take the gene from a functioning person, a person who makes insulin. We can take a bacterial cell, and in a bacterial cell they have a section of DNA called a plasmid, We can snip that plasmid open, so you can see it's almost like a 3-ring binder. We've popped it open, and we can take this gene from a human cell that produces insulin. We can take that gene and we can stitch it, glue it in place here into this plasmid. We can then take this plasmid that now contains the human gene for insulin, put it back inside of a bacteria. That bacteria will replicate over and over and over again, making billions and billions of bacterial cells that each have that human gene for insulin in it. So that's how we get our insulin produced today. Another example of this with gene therapy would be for cystic fibrosis. So somebody who has cystic fibrosis, we can take a cell from a person who doesn't have cystic fibrosis, has the normal gene for producing normal mucus in the respiratory system. We can remove that gene. We can take a virus, so this could be a pneumonia, it could be any type of virus that would typically infect the chest, common cold. We take the bad genetics out of it, the stuff that causes it to be pneumonia or whatever the disease is. We take those bad genes away because we don't want to infect a person with the disease. And we insert that human gene in place. When the person inhales that virus, we can put this in, load this up into like a puffer for Ventolin. People who take for asthmatics take, person would puff that in along with a whole bunch of other viral particles. Again, they don't have the virus information inside of them, they just have that human gene inside. And with some success, when that virus injects its genetic information inside the host cell, that host cell can take up sometimes that genetic information, and there has been some success with this where the person will no longer have cystic fibrosis. Now the main issue with this, of course, in science is ethical issues. And there are a whole bunch of ethics topics in your textbook that'll be good things for you to look at, to describe, or to discuss, and think about what are the benefits of some of these things. Looking at labeling of food by food manufacturers. Should we modify our genetics? If we can, should it be something that we're allowed to do, or should we be modifying other organisms?